Last spring, I watched my neighbor lose an entire crop of tomatoes and peppers, not from pests or disease, but from something far more sinister. His black walnut tree was silently poisoning the soil with juglone, a chemical compound that creates toxic conditions for nightshade plants within a 50 to 80-foot radius. What he didn't realize is that allelopathy, the scientific term for plant chemical warfare, operates at the molecular level, disrupting photosynthesis and triggering oxidative stress that literally suffocates neighboring crops. The invisible chemical assault was happening beneath his feet for weeks, yet he kept searching above ground for visible threats that simply weren't there. This chemical warfare operates through allelopathy, a biochemical system where plants manufacture their own herbicides to eliminate competition. Secondary metabolites function as precision-engineered toxins, targeting specific cellular processes in neighboring plants with lethal efficiency. The chemical arsenal deployed through allelopathy systematically dismantles the fundamental machinery that sustains plant life. These compounds simultaneously attack photosynthesis, root development, and nutrient uptake mechanisms, creating cascading failures throughout the target plant's physiological systems. The process begins when allelopathic plants release phenolic compounds, terpenoids, and alkaloids through multiple pathways. Root exudates seep directly into soil. Decomposing leaf matter releases concentrated toxins, and volatile compounds disperse through air currents to contaminate surrounding growing areas. Research demonstrates that phenolic compounds at specific concentrations trigger reactive oxygen species bursts within target plant cells. These molecular reactions initiate membrane lipid peroxidation, a chain reaction that destroys the protective barriers surrounding cellular components. Meanwhile, the same compounds interfere with crucial enzyme functions, blocking the biochemical pathways that convert sunlight into usable energy and nutrients into cellular building blocks. The toxicity operates on a concentration gradient that determines whether neighboring plants experience minor growth reduction or complete physiological collapse. Low-level exposure may only slow development rates and reduce yield potential, while higher concentrations trigger immediate cellular damage that progresses rapidly to plant death. The concentration-dependent nature of allelopathic toxicity creates invisible zones of varying lethality around each chemically aggressive plant. These chemical compounds persist in soil matrices for weeks or months after release, creating toxic zones that extend far beyond the visible canopy or root system of the source plant. The persistence depends on soil composition, drainage patterns, and microbial activity. Clay soils retain compounds longer, while sandy soils allow faster dispersal and dilution. Organic matter can either accelerate decomposition of some toxins or bind with others to extend their active lifespan in the growing medium. The molecular mechanisms of allelopathic damage target multiple cellular systems simultaneously. Compounds inhibit photosystem II, the protein complex responsible for capturing light energy and converting it into chemical energy. Other toxins disrupt mitochondrial function, effectively starving cells of the ATP molecules needed for basic metabolic processes. Cell membrane integrity becomes compromised as allelopathic compounds alter lipid composition and protein function, causing cellular contents to leak out while preventing essential nutrients from entering. The delayed response characteristic of chemical warfare creates the most insidious aspect of allelopathic damage. Target plants may maintain normal appearance for days or weeks, while toxic compounds gradually infiltrate their tissues and accumulate to lethal concentration. This time lag between exposure and visible symptoms makes diagnosis extremely challenging. By the time wilting, chlorosis, or stunted growth becomes apparent, extensive internal damage has already occurred. Terpenoids represent another major class of allelopathic compounds that operate through different molecular pathways than phenolic acids. These volatile organic compounds can cross cell membranes more easily, allowing rapid penetration into target tissues where they disrupt protein synthesis and enzyme function. Alkaloids provide yet another chemical weapon. Ibistai, interfering with nitrogen metabolism and blocking the formation of essential amino acids needed for cellular repair and growth. The systematic nature of allelopathic attack explains why affected plants often display multiple symptoms simultaneously. Yellowing leaves indicate disrupted chlorophyll production. Wilting suggests compromised water transport systems, and stunted root development reveals blocked cell division processes. These compound effects distinguish chemical warfare from single-factor problems like nutrient deficiencies or specific pest damage. Soil conditions dramatically influence the effectiveness and persistence of allelopathic compounds. Well-drained soils with active microbial populations can decompose some toxins relatively quickly, while waterlogged or sterile growing media may preserve chemical activity for extended periods. Temperature fluctuations affect both the release rate of compounds from source plants and their degradation rate in soil. Understanding the molecular basis of plant chemical warfare reveals why certain crop combinations consistently fail, despite optimal soil fertility, adequate irrigation, and proper pest management. The invisible toxic zones created by allelopathic compounds operate independently of traditional growing factors, creating failure patterns that appear random to gardeners unfamiliar with these biochemical interactions. The precision of allelopathic targeting often affects specific plant families while leaving others completely unharmed. This selective toxicity results from differences in cellular structure, metabolic pathways, and detoxification mechanisms between plant species. These biochemical vulnerabilities determine which crops will thrive, struggle, or die when exposed to specific allelopathic compounds in the surrounding soil matrix. While many plants engage in chemical warfare, certain species have evolved particularly devastating arsenals that can transform entire growing areas into sterile wastelands where nothing else survives. Among all allelopathic plants documented by researchers, black walnut demonstrates the most devastating chemical warfare capabilities, transforming productive garden areas into sterile dead zones that persist for years. These majestic trees function as biological weapons factories, manufacturing juglone, a naphthoquinone compound that accumulates throughout every tissue of the tree, including roots, bark, leaves, and protective hulls surrounding the nuts. The juglone production system operates continuously throughout the growing season, releasing concentrated toxins through multiple pathways that contaminate surrounding soil matrices. Rainfall dissolves juglone from leaf surfaces and bark crevices, carrying the dissolved compound directly into root zones of neighboring plants. Decomposing leaf litter creates concentrated toxic hotspots as cellular breakdown releases stored juglone in concentrated doses that leach into underlying soil layers. Root exudation provides the most persistent delivery mechanism, with black walnut roots actively secreting juglone directly into the soil solution, where it binds with organic matter and clay particles. The continuous underground chemical release creates stable toxic concentrations that maintain lethal levels throughout multiple growing seasons, effectively sterilizing large areas around each tree. The compound demonstrates extraordinary persistence in soil environments, maintaining active toxicity for extended periods after complete tree removal. This chemical contamination means that gardeners who remove problematic black walnut trees may continue experiencing crop failures for multiple growing seasons before soil conditions return to normal. The persistent nature results from juglone's molecular structure, which resists breakdown by soil microorganisms and remains stable across varying temperature and moisture conditions. Juglone toxicity operates far beyond the visible tree canopy, creating invisible zones of chemical contamination that extend 50 to 80 feet from mature specimens, though this range varies significantly with soil type. The drip line represents only the beginning of the toxic zone. The highest concentrations occur directly beneath the canopy, but lethal levels persist much further outward than most gardeners anticipate. Sandy soils allow rapid lateral movement, while clay soils concentrate the compound in smaller, more intensely toxic areas. The molecular mechanism of juglone toxicity targets the electron transport chain within plant mitochondria, the cellular powerhouses responsible for energy production. When juglone molecules enter plant cells, they disrupt the biochemical machinery that converts nutrients and oxygen into usable cellular energy. This disruption cascades through all cellular functions, since energy production provides the foundation required for photosynthesis, protein synthesis, and cellular repair mechanisms. Susceptible plants display rapid symptom progression that distinguishes juglone poisoning from other garden problems. Affected plants quickly begin exhibiting characteristic wilting despite adequate soil moisture levels. The wilting occurs because juglone disrupts water transport mechanisms in plant vascular systems, preventing proper hydration even when roots remain surrounded by moist soil. Leaf yellowing follows as chlorophyll production systems shut down under the chemical assault. The yellowing typically begins with younger leaves and progresses throughout the plant as juglone concentrations build up in plant tissues. Root development becomes severely stunted, with new root tips turning black and dying as they encounter toxic soil zones, preventing the plant from accessing nutrients and water needed for recovery. Research measurements reveal that juglone concentrations as low as 25 parts per million inhibit seed germination in susceptible species, demonstrating the compound's effectiveness at preventing competitor establishment. Higher concentrations approaching 100 parts per million cause immediate cellular membrane damage, triggering rapid plant death through multiple simultaneous system failures. The nightshade family faces particular vulnerability to juglone exposure due to their cellular structure and metabolic characteristics. Tomatoes, peppers, eggplants, and potatoes possess high metabolic rates that accelerate juglone uptake and accumulation, causing these valuable crops to concentrate toxic levels more rapidly than resistant species. When planted within the contamination zone, nightshade plants typically display symptoms rapidly and progress to complete plant death within days. Other susceptible crops include members of the rose family, many annual flowers, and most vegetable crops commonly grown in home gardens. Apple trees, cherry trees, and other fruit trees show particular sensitivity, often declining gradually over several growing seasons when planted too close to black walnut specimens. The juglone system represents allelopathic chemical warfare at its most refined level, demonstrating how a single compound can systematically eliminate entire plant communities from the surrounding ecosystem. The chemical operates with pharmaceutical precision, targeting specific metabolic pathways while maintaining stable concentrations that prevent adaptation or tolerance development in susceptible species. This biochemical dominance allows black walnut trees to maintain clear zones around their trunks, eliminating competition for water, nutrients, and light through chemical rather than physical means. The systematic nature of juglone toxicity creates predictable patterns of plant success and failure, making black walnut the primary example of why distance calculations and species selection require scientific precision rather than guesswork. Understanding juglone's reach and persistence allows gardeners to make informed decisions about plant placement, preventing costly crop failures through strategic positioning that respects the invisible but deadly chemical boundaries these trees establish. Yet among all the plants vulnerable to these chemical attacks, certain crop families demonstrate such extreme sensitivity that they serve as the first indicators when toxic compounds infiltrate growing areas. The Solanaceae family represents the most vulnerable target in allelopathic warfare, transforming our most prized garden crops into the first casualties when chemical compounds infiltrate growing areas. Tomatoes, peppers, eggplants, and potatoes share cellular architecture that makes them exceptionally susceptible to phytotoxic assault. Their very design for rapid growth and high productivity becomes their greatest weakness when facing molecular attack. The accelerated metabolic machinery that drives nightshade vigor operates like a high-performance engine running at maximum capacity. These plants maintain elevated rates of cellular respiration, protein synthesis, and nutrient transport to support their rapid development and heavy fruit production. The same biological systems that enable nightshades to produce abundant harvests also accelerate the absorption and concentration of allelopathic compounds throughout their tissues. Cell division processes in nightshade plants occur at frequencies that dwarf most other crop families. This rapid cellular multiplication requires continuous energy production and raw material processing, creating multiple entry points where toxic compounds can infiltrate and disrupt essential biochemical pathways. The high metabolic demand forces nightshade cells to maintain maximum permeability for nutrient uptake, inadvertently providing easy access for chemical warfare agents. When juglone or similar allelopathic compounds contact nightshade root systems, the disruption begins immediately at the chlorophyll synthesis level. The molecular machinery responsible for constructing chlorophyll molecules becomes compromised as toxic compounds interfere with the enzyme systems that assemble the complex porphyrin ring structures essential for photosynthesis. This disruption manifests as characteristic yellowing that appears rapidly after initial contact. The chlorosis pattern typically begins with the youngest, most metabolically active leaves, before spreading throughout the plant as compound concentrations build up in vascular tissues. Unlike nutrient deficiency yellowing that follows predictable patterns based on element mobility, allelopathic chlorosis often appears random and asymmetrical, reflecting the uneven distribution of toxic compounds as they move through plant tissues. Water regulation systems face simultaneous assault as allelopathic compounds target the specialized proteins that control cellular water balance. The compounds interfere with aquaporins, the channel proteins that regulate water movement across cell membranes, causing disrupted hydration even when soil moisture remains adequate. Root tissue damage compounds the problem by reducing the surface area available for water absorption and creating physical barriers to efficient water transport. The resulting wilting symptoms frequently mislead gardeners into diagnosing drought stress or root rot problems. Plants may receive additional irrigation or fungicide treatments that provide no benefit while the true chemical assault continues unabated. The wilting progresses rapidly as vascular transport systems become increasingly compromised by accumulating toxins. Mitochondrial function becomes severely impaired as allelopathic compounds penetrate cellular power plants and disrupt ATP synthesis pathways. These organelles normally operate like biological batteries, converting nutrients and oxygen into usable energy through complex electron transport chains. Toxic compounds intercept electrons during these crucial energy conversion processes, effectively short-circuiting the cellular systems that power all metabolic functions. The energy crisis cascades through every aspect of plant physiology as ATP levels plummet. Protein synthesis slows dramatically when cells lack sufficient energy to drive the complex molecular assembly processes. Cellular repair mechanisms shut down as available. Energy gets diverted to emergency survival functions. Growth ceases as expanding tissues require massive energy investments that compromised mitochondria cannot provide. Oxidative stress represents another devastating consequence of allelopathic exposure in nightshade plants. The compromised cellular systems begin producing excessive reactive oxygen species, highly unstable molecules that attack cell membranes, proteins, and DNA through aggressive oxidation reactions. These molecular reactions create a cascade of cellular damage that spreads throughout plant tissues. Cell membrane integrity deteriorates rapidly under oxidative assault, allowing cellular contents to leak out while preventing essential nutrients from entering. DNA damage accumulates as reactive oxygen species attack the genetic material responsible for directing cellular functions and repair processes. The combination of energy starvation and oxidative damage creates a lethal synergy that accelerates plant decline. Even plants that survive the initial chemical assault face long-term consequences that extend far beyond visible symptoms. Fruit production declines dramatically as energy resources get redirected from reproductive functions to cellular survival and repair. The fruits that do develop often exhibit delayed maturity, reduced size, and compromised quality as the plant struggles to complete normal developmental processes under chemical stress. Disease resistance mechanisms become severely compromised in allelopathic stressed nightshade plants. The energy crisis prevents proper immune system function, while oxidative damage impairs the cellular systems responsible for producing defensive compounds. Weakened plants become vulnerable to secondary infections that would normally pose no threat to healthy specimens. The extreme sensitivity of nightshade crops transforms them into biological early warning systems for allelopathic soil contamination. Their rapid response to chemical warfare agents provides gardeners with immediate feedback about soil toxicity levels before less sensitive crops show any symptoms. A thriving tomato plant can shift from apparent health to obvious distress rapidly after encountering allelopathic compounds. Strategic placement of nightshade test crops allows systematic mapping of toxic zones around suspected allelopathic plants. The concentration-dependent response patterns help determine safe planting distances while identifying areas where soil remediation may be necessary. Understanding these biochemical vulnerabilities enables gardeners to position valuable nightshade harvests beyond the reach of chemical warfare while using their sensitivity as a diagnostic advantage. Yet nightshades represent just one category of vulnerable crops. Others seemingly innocent garden plants deploy their own sophisticated chemical arsenals, targeting different metabolic pathways with precision that creates entirely different patterns of agricultural devastation. The golden faces that brighten summer gardens conceal one of nature's most sophisticated chemical warfare operations, deploying precision-engineered molecular weapons that systematically eliminate specific crop families while leaving others completely untouched. Sunflowers manufacture allelopathic compounds that accumulate throughout their cellular tissues, creating concentrated chemical arsenals within leaves, stems, and root systems that persist long after the spectacular blooms fade. The chemical production system operates continuously throughout the growing season, with compound concentrations building steadily as plants mature and expand their biomass. These allelopathic molecules concentrate most heavily in leaf tissues and stem structures, where they await release through natural decomposition processes and active root exudation into surrounding soil matrices. The timing creates maximum impact. Peak toxin release occurs when neighboring crops are establishing root systems and beginning rapid growth phases. Unlike broad-spectrum herbicides that damage all plant life indiscriminately, sunflower compounds target specific metabolic pathways with surgical precision. The selective nature of this chemical assault creates a biological sorting system where certain plant families face immediate devastation, while others grow normally just inches away. This targeting specificity results from molecular recognition systems. The allelopathic compounds bind preferentially to particular enzyme structures and cellular receptors found only in susceptible species. Root exudates provide the primary delivery mechanism for sunflower toxins, releasing steady streams of phenolic compounds directly into the soil solution where they bind with organic matter particles and clay minerals. This binding action creates stable chemical complexes that resist degradation by soil microorganisms, extending the active lifespan of toxic compounds far beyond the sunflower growing season. The persistence factor transforms temporary chemical warfare into long-term soil contamination that affects subsequent crop rotations. Research demonstrates that sunflower residues significantly reduce germination rates in sensitive crops, with inhibitory effects persisting for months after harvest and residue incorporation. The compound stability in soil environments means that farmers planting susceptible crops in fields previously occupied by sunflowers face predictable establishment failures despite optimal growing conditions and proper management practices. The molecular mechanism targets protein synthesis pathways in susceptible plants, effectively sabotaging the cellular machinery responsible for manufacturing essential enzymes. This biochemical disruption prevents the formation of critical proteins needed for photosynthesis, nutrient transport, and cellular repair processes. Without functional enzyme systems, affected plants cannot maintain basic metabolic functions even when provided with adequate water, nutrients, and sunlight. The protein synthesis disruption cascades through multiple physiological systems simultaneously. Photosynthetic enzyme production ceases, preventing chlorophyll formation and blocking the conversion of sunlight into usable energy. Transport proteins become unavailable, disrupting the movement of nutrients and water throughout plant tissues. Repair enzymes cannot be synthesized, leaving damaged cellular components unable to recover from normal environmental stresses. Nightshade crops demonstrate exceptional vulnerability to sunflower chemical warfare due to their specialized cellular architecture and rapid metabolic rates. The allelopathic compounds specifically interfere with the high-energy cellular processes that these plants require for vigorous growth and fruit production. This disruption eliminates the nightshade's competitive advantages while compromising their ability to complete normal developmental cycles. The chemical interference prevents proper cellular function in tomatoes, peppers, eggplants, and potatoes by targeting the same metabolic pathways that make these crops so productive under normal conditions. The rapid cellular division and high nutrient transport rates that characterize healthy nightshade growth become vulnerabilities when allelopathic compounds infiltrate plant tissues and disrupt essential biochemical processes. Small grain crops and members of the brassica family also suffer significant yield reductions when planted in rotation sequences following sunflowers. Wheat, barley, and oat establishment becomes compromised as allelopathic residues interfere with seed germination and early seedling development. The timing proves particularly damaging since grain crops require rapid establishment and early season vigor to maximize yield potential. Brassica crops including cabbage, broccoli, and canola demonstrate similar sensitivity patterns, experiencing reduced germination rates and stunted early growth when planted in sunflower-contaminated soils. The compound persistent means that even delayed planting schedules cannot avoid the chemical interference. Toxic levels remain active throughout multiple growing seasons following sunflower cultivation. The selective toxicity creates predictable agricultural patterns that allow experienced farmers to anticipate rotation failures before planting decisions are finalized. Fields previously planted to sunflowers consistently underperform when planted to susceptible crops, regardless of soil fertility levels, moisture conditions, or pest management practices. The chemical warfare operates independently of traditional growing factors, creating yield limitations that persist until soil detoxification occurs naturally through compound degradation. Commercial agriculture has documented these interaction patterns extensively, revealing consistent crop performance data that correlates directly with sunflower cultivation history rather than environmental variables. The chemical fingerprint left by sunflower allelopathy creates measurable yield impacts that extend across multiple crop species and growing seasons. The concentration-dependent nature of sunflower toxicity means that plant density and residue management practices directly influence the severity of subsequent crop damage. Dense sunflower stands produce higher total chemical loads, while residue incorporation accelerates compound release into soil systems. Understanding these variables allows strategic management of sunflower cultivation to minimize long-term soil contamination while maintaining productive crop rotations. This biochemical dominance explains why certain rotation sequences consistently fail to meet yield expectations despite seemingly optimal growing conditions and management inputs. Yet the relationship between chemical concentration and plant response reveals even more sophisticated patterns where the same compounds can produce dramatically different effects depending on their concentration levels in the surrounding environment. Allelopathic warfare reveals its most sophisticated dimension through concentration-dependent chemical systems that transform identical molecular compounds from growth enhancers into lethal toxins based purely on dosage levels. These systems create invisible gradients of biological activity radiating outward from chemical source plants, like ripples in a biochemical pond, where the same compounds produce dramatically different effects depending on their concentration in the surrounding environment. The concentration gradient phenomenon creates distinct biological zones around allelopathic plants where identical species experience dramatically different outcomes based solely on their distance from the chemical source. Research demonstrates that allelopathic compounds at low concentrations actually stimulate cellular activity in many plant species, enhancing root elongation rates and photosynthetic efficiency through mild biochemical activation of growth processes. These beneficial effects operate through hormetic responses, where low-level chemical stress triggers enhanced cellular performance and improved stress resistance mechanisms. The stimulation occurs as plants activate their natural detoxification systems in response to mild chemical exposure, creating enhanced metabolic efficiency that translates into improved growth rates and vigor. Root systems exposed to these low concentrations develop increased surface area and enhanced nutrient absorption capacity compared to plants grown in chemical-free environments. Photosynthetic enhancement at low compound concentrations results from mild activation of chlorophyll synthesis pathways and improved electron transport efficiency within photosystem complexes. The chemical stimulation optimizes light harvesting mechanisms and carbon fixation processes, allowing plants to extract more energy from available sunlight while maintaining normal cellular functions. These performance improvements demonstrate that many allelopathic compounds prove beneficial at low doses while becoming toxic at higher concentrations. The critical threshold occurs when chemical concentrations cross biological tipping points, where the same compounds that previously enhanced growth begin triggering oxidative stress responses and cellular damage cascades. The transition from beneficial to harmful effects happens rapidly as compound levels exceed plant detoxification capacity, transforming growth stimulants into cellular toxins through simple concentration increases. Plants that thrived under mild chemical exposure experience immediate physiological distress when compound levels surpass their tolerance thresholds. Oxidative stress responses manifest as reactive oxygen species production increases beyond cellular antioxidant capabilities, creating molecular damage that spreads throughout plant tissues. Cell membrane integrity becomes compromised as lipid peroxidation reactions destroy the protective barriers surrounding cellular components, allowing toxic compounds to penetrate deeper into sensitive cellular structures. The cascade effect accelerates as damaged cells lose their ability to regulate chemical uptake and detoxification processes. The spatial distribution of these concentration effects creates concentric zones of biological activity around allelopathic plants, each supporting different plant communities based on their chemical tolerance thresholds. Plants requiring growth stimulation established successfully at intermediate distances, where compound concentrations remain within beneficial ranges. Species sensitive to chemical exposure must establish beyond the outer edges of the concentration gradient to avoid toxic effects entirely. This zonation pattern explains the seemingly random success and failure patterns observed in allelopathic-dominated landscapes, where identical plant species planted at different distances experience completely different outcomes. Plants positioned at optimal distances within the stimulation zone often outperform those grown in chemical-free areas, while specimens placed too close to allelopathic sources suffer rapid decline despite identical care and growing conditions. The dose-response relationship demonstrates that allelopathy operates according to pharmacological principles rather than simple toxicity models, where the difference between beneficial and harmful effects depends entirely on chemical concentration rather than compound identity. This relationship reveals why distance calculations become critical for successful plant establishment near allelopathic species, requiring precise positioning to capture beneficial effects while avoiding toxic exposure level. Understanding concentration gradients allows strategic garden planning that maximizes the beneficial aspects of allelopathic interactions while preventing chemical damage to valuable crops. Plants can be positioned at distances where they receive mild chemical stimulation that enhances their performance without crossing the threshold into toxicity ranges that trigger cellular damage and growth inhibition. The concentration-dependent nature of chemical warfare creates predictable patterns of plant success and failure that extend far beyond the dramatic examples of black walnut and sunflower toxicity. Even the most common plants deploy sophisticated chemical arsenals that operate through these same principles, creating invisible boundaries that determine which species can establish and thrive in any given location. These chemical systems operate continuously beneath our feet, establishing dominance patterns that shape entire plant communities through molecular precision rather than visible competition. The most ubiquitous examples of this warfare remain hidden in plain sight, deployed by plants so common that their chemical influence goes completely unrecognized. Fennel demonstrates how common garden herbs deploy sophisticated chemical arsenals that systematically eliminate competing plant species through precision molecular warfare. This aromatic Mediterranean native manufactures allelopathic compounds that create exclusion zones around each plant, preventing seed germination and seedling establishment in most other species while maintaining its own dominance across growing areas. The chemical production system operates continuously throughout the growing season, with fennel plants releasing steady streams of allelopathic compounds through both root exudation and volatile emissions from above-ground tissues. These compounds accumulate in soil matrices and disperse through air currents, creating multiple delivery pathways that ensure comprehensive chemical coverage of the surrounding environment. The timing maximizes competitive advantage. Peak toxin release occurs during spring establishment periods when potential competitors attempt germination and early development. Fennel's allelopathic compounds target fundamental cellular processes in susceptible plants, disrupting protein synthesis pathways essential for normal growth and development. The molecular interference prevents the formation of critical enzymes needed for photosynthesis, nutrient transport, and cellular repair mechanisms. Without functional enzyme systems, affected plants cannot maintain basic metabolic functions even when provided with adequate water, nutrients, and optimal growing conditions. The chemical warfare system demonstrates remarkable selectivity, affecting some plant families while leaving others completely unharmed. This targeting specificity results from molecular recognition systems, where allelopathic compounds bind preferentially to particular cellular structures found only in susceptible species. The selective nature creates biological sorting systems where certain crops face immediate establishment failure, while others grow normally just inches away from fennel plants. Root system development faces particular vulnerability to fennel's chemical assault, as the compounds concentrate in soil zones where germinating seeds must establish their primary root network. The allelopathic interference disrupts root tip growth and cellular division processes, preventing the formation of functional root systems needed for water and nutrient absorption. Seeds that successfully germinate often fail to develop adequate root systems, leading to establishment failure despite favorable environmental conditions. The concentration-dependent effects create distinct zones of biological activity around fennel plants, where identical species experience dramatically different outcomes based solely on their distance from the chemical source. Plants positioned at intermediate distances may experience mild growth suppression, while those planted too close suffer complete establishment failure or rapid decline after initial growth appears normal. Chemical persistence extends fennel's dominance far beyond the active growing season, as allelopathic compounds remain stable in soil environments for extended periods following their release. Even after complete fennel removal through mechanical means, soil toxicity levels can prevent successful crop establishment for weeks or months until natural degradation processes reduce compound concentrations below harmful thresholds. The persistence depends on soil organic matter content, microbial activity levels, and environmental conditions that influence chemical breakdown rates. This extended chemical contamination explains why gardeners attempting to establish diverse plantings in areas previously occupied by fennel often experience mysterious establishment failures despite proper soil preparation and favorable growing conditions. The invisible chemical residues continue operating as biological herbicides long after the fennel plants responsible for their production have been eliminated from the site. Soil testing typically reveals adequate fertility and proper pH levels, masking the true cause of establishment problems. The molecular mechanisms demonstrate that allelopathic compounds function according to pharmacological principles, where the difference between mild growth suppression and lethal toxicity depends entirely on chemical concentration rather than compound identity. This relationship reveals why distance calculations become critical for successful plant establishment near allelopathic species, requiring precise positioning to avoid toxic exposure levels that trigger cellular damage and growth inhibition. Understanding fennel's chemical warfare capabilities allows strategic garden planning that prevents costly crop failures through informed plant placement decisions. Soil remediation techniques can accelerate the neutralization of residual compounds, including activated carbon applications that bind with allelopathic molecules and organic matter additions that support increased microbial activity for faster natural degradation. The recognition of common herb allelopathy transforms garden planning from random plant placement into strategic positioning that accounts for invisible chemical boundaries and compound persistence patterns. These chemical systems operate continuously beneath the surface, establishing dominance patterns that determine which species can successfully establish and thrive in any given location. The widespread nature of allelopathic interactions means that chemical warfare influences plant communities far more extensively than most gardeners realize, creating invisible patterns of success and failure that appear random without understanding the underlying molecular mechanisms. Yet when these chemical assaults begin affecting valuable crops, the resulting symptoms follow distinctive patterns that reveal the true nature of the biological warfare occurring beneath the soil surface. Recognizing these chemical attacks requires understanding the distinctive physiological signatures that differentiate allelopathic warfare from conventional garden problems. Chemical compound interference creates unique cellular responses that follow predictable pathways, generating symptom clusters that distinguish molecular damage from nutrient deficiencies, pest attacks, or pathogenic infections. The diagnostic challenge intensifies because allelopathic damage mimics common cultivation problems, leading gardeners to misinterpret chemical warfare symptoms as irrigation issues or soil fertility imbalances. These misdiagnoses trigger counterproductive treatment applications that fail to address the underlying toxicity while potentially exacerbating cellular stress through additional chemical inputs. Plants suffering from allelopathic exposure receive unnecessary fertilizer application, modified watering schedules, or fungicide treatments that provide no relief while masking the true source of physiological decline. Intervenal chlorosis emerges as the primary visual indicator of allelopathic interference, manifesting as distinctive yellowing patterns between leaf veins while the vascular structures themselves retain normal green coloration. This specific symptom pattern results from disrupted chlorophyll synthesis pathways, where allelopathic compounds interfere with the cellular machinery responsible for constructing light-harvesting pigments. The selective nature of this chlorosis distinguishes chemical damage from nutrient deficiency symptoms, which typically produce uniform yellowing patterns or affect older leaves, preferentially as mobile nutrients relocate to support new growth. The chlorophyll disruption operates where allelopathic agents block key enzymes involved in pigment formation, the complex molecular structures that capture light energy during photosynthesis. This biochemical interference prevents normal chloroplast function while leaving the vascular framework intact, creating the characteristic pattern where leaf veins appear as green highways traversing yellow cellular landscapes. The timing of symptom appearance correlates directly with compound exposure levels and plant sensitivity threshold. Wilting symptoms develop simultaneously despite adequate soil moisture availability, as allelopathic compounds systematically disrupt water uptake and transport mechanisms throughout root and stem vascular systems. The molecular interference targets specialized proteins that regulate water movement across cellular membranes, preventing normal hydration processes even when plant roots remain surrounded by moist, growing medium. This chemical disruption of water relations creates the paradoxical situation where plants display drought stress symptoms while growing in properly irrigated soil conditions. The water transport disruption compounds rapidly as allelopathic agents damage root cell membranes and interfere with the pressure systems that drive water movement from soil through plant tissues. Vascular integrity becomes compromised as toxic compounds accumulate within transport tissues, blocking the mechanisms that maintain cellular turgor pressure and structural support. Plants experience systemic dehydration despite continuous water availability, leading to characteristic wilting that resists correction through increased irrigation applications. Root system development reveals the most diagnostic evidence of allelopathic attack, with affected plants producing stunted, discolored underground structures that display blackened tips, where chemical concentrations reach maximum soil solution levels. The root tip necrosis occurs because actively dividing cellular regions responsible for root elongation face direct exposure to concentrated allelopathic compounds as they penetrate soil matrices. These growing points possess high metabolic rates and cellular permeability that accelerate toxin uptake and accumulation. The root damage progresses systematically as chemical compounds interfere with cell division processes essential for tissue expansion and organ development. Primary root systems fail to establish proper structural architecture, while secondary root formation becomes severely limited as cellular machinery suffers continued chemical assault. The compromised root networks cannot provide adequate nutrient and water absorption capacity, creating cascading physiological stress that extends throughout the entire plant system. Growth asymmetry develops as proximity to allelopathic sources determines chemical exposure intensity, creating distinctive damage gradients where plant structures closest to toxin sources exhibit severe symptoms while distant tissues maintain relatively normal appearance. This spatial pattern distinguishes allelopathic damage from systemic problems like viral infections or nutritional imbalances that affect plants uniformly. The gradient effect demonstrates the concentration-dependent nature of chemical warfare, where distance determines survival probability. The asymmetrical damage patterns extend beyond individual plants to affect entire garden beds, creating zones of varying plant health that correlate directly with distance from allelopathic sources rather than traditional growing factors like soil fertility or drainage patterns. Plants positioned within high concentration zones display rapid decline, while identical specimens located beyond the chemical influence boundaries thrive under otherwise identical cultivation conditions. Reproductive failure emerges as the most economically devastating consequence of allelopathic exposure, with surviving plants experiencing dramatic reductions in flowering and fruit production as cellular energy reserves redirect toward survival and repair mechanisms rather than reproductive development. Even plants that appear to recover from initial chemical assault continue suffering reduced yield potential as metabolic resources prioritize cellular maintenance over grain, fruit, or seed production. Accurate symptom recognition enables rapid diagnostic differentiation between allelopathic damage and conventional plant health problems, allowing immediate implementation of corrective strategies. Understanding these warning signs transforms mysterious crop failures into manageable situations where strategic positioning decisions can prevent chemical warfare before it devastates valuable plantings. Establishing safe distances between allelopathic plants and susceptible crops demands precise understanding of how chemical compounds migrate through soil matrices and concentrate within root zone environments. Chemical dispersal operates according to predictable physical and biological principles that govern molecule movement, soil binding, and compound persistence across different growing environments. These scientific patterns enable gardeners to calculate minimum separation distances based on soil characteristics, drainage conditions, and specific plant physiology rather than relying on generalized spacing recommendations that frequently underestimate chemical reach. Traditional garden spacing guidelines consistently underestimate the true extent of allelopathic compound dispersal, creating false security that leads to devastating crop failures even when plants appear positioned at safe distances. Standard recommendations typically focus on visible canopy spread or estimated root zone diameter, ignoring the invisible chemical contamination zones that extend far beyond physical plant boundaries. These miscalculations result in systematic crop losses as toxic compounds infiltrate supposedly protected growing areas and accumulate to lethal concentrations in sensitive plant root systems. Juglone dispersal from black walnut trees demonstrates the extreme distances required for complete chemical safety, with buffer zones extending 50 to 80 feet from mature specimens to ensure reliable protection for highly sensitive crops. These measurements represent minimum distances under optimal soil conditions. Certain soil types and environmental factors can extend chemical reach even further. The extensive reach reflects juglone's molecular stability and persistence in soil environments, where the compound maintains toxic activity across vast areas and extended time periods. Soil drainage characteristics dramatically influence allelopathic compound movement patterns and concentration distributions throughout growing areas. Well-drained soils facilitate chemical dispersal and dilution as water movement carries dissolved compounds away from source plants and distributes them across larger soil volumes. Poor drainage creates concentrated chemical accumulation zones where compounds remain trapped in smaller areas for extended periods, maintaining higher toxicity levels within restricted zones around source plants. Elevation changes within growing areas generate predictable compound flow patterns that create accumulation zones where sensitive plants face heightened chemical exposure risks. Allelopathic compounds dissolved in soil water follow gravity-driven drainage patterns, flowing downhill and concentrating in low-lying areas where natural depressions collect runoff water. These accumulation zones maintain elevated compound concentrations that persist longer than surrounding areas, creating toxic hotspots that prove especially dangerous for susceptible crop species. The concentration effect intensifies during rainfall events when water movement accelerates chemical transport from source plants to collection areas. Storm water runoff carries dissolved allelopathic compounds from hillside positions to valley floors, creating temporary but highly concentrated toxic conditions in areas that might otherwise remain safe for sensitive plant cultivation. Understanding these flow patterns allows strategic positioning of valuable crops in elevated areas that remain above the chemical accumulation zones. Soil amendment strategies can significantly reduce allelopathic compound mobility and create opportunities for closer plant spacing in properly managed growing environments. Activated carbon applications bind directly with allelopathic compounds, neutralizing their biological activity and reducing soil toxicity levels within treated zone. The carbon binding creates stable complexes that prevent compound migration while accelerating natural degradation processes through enhanced microbial activity. Organic matter additions support increased microbial populations that accelerate allelopathic compound breakdown through enhanced biological degradation processes. The microbial activity creates enzyme systems capable of metabolizing toxic compounds into harmless byproducts, reducing chemical persistence and shortening the time required for soil detoxification. Strategic compost applications and green manure incorporation can significantly reduce buffer zone requirements through accelerated compound neutralization. Wind patterns affect volatile compound dispersal from aromatic plants, creating directional contamination patterns that require consideration of prevailing air movements when positioning sensitive crop species. Volatile allelopathic compounds travel considerable distances through air currents before settling onto soil surfaces and penetrating root zones of susceptible plants. The directional nature of wind dispersal creates asymmetrical contamination zones where downwind areas face higher chemical exposure risk than upwind positions. Seasonal wind patterns create predictable contamination directions that allow strategic plant positioning to minimize chemical exposure risks. Understanding local meteorological patterns enables gardeners to position sensitive crops in locations where prevailing winds carry volatile compounds away from rather than toward valuable plant stands. The seasonal variation in wind direction requires consideration of year-round air movement patterns rather than temporary weather conditions. Buffer zone calculations based on scientific compound dispersal principles transform random plant placement strategies into precision positioning systems that eliminate toxic interactions while maximizing productive use of available growing space. These calculated separation distances account for soil type, drainage patterns, elevation changes, and atmospheric conditions to create reliable protection zones around allelopathic plants. The systematic approach prevents chemical warfare through strategic spatial management that maintains safe growing conditions for susceptible crop species while optimizing garden productivity across the entire cultivation area. The transformation from guesswork to calculated precision reveals how molecular interactions determine success or failure in every garden bed, where invisible chemical boundaries operate with the same predictable accuracy as any pharmaceutical compound. Mastering these molecular interactions transforms cultivation from experimental guesswork into calculated biochemistry, where strategic plant positioning harnesses beneficial chemical stimulation while avoiding the toxic compound concentrations that trigger crop annihilation. The key practical takeaways center on precision distance calculations, maintaining 50 to 80-foot buffer zones around black walnut trees, and understanding that concentration gradients determine whether identical compounds enhance or destroy plant growth. These invisible molecular battles follow predictable patterns based on soil drainage, compound persistence, and concentration gradients that extend far beyond visible plant boundaries. Successful gardeners apply activated carbon treatments to neutralize soil toxicity and position sensitive crops beyond chemical warfare zones, rather than relying on traditional spacing guidelines that consistently underestimate allelopathic reach. What plant combination has devastated your harvest, and how will you redesign your cultivation strategy to prevent future biochemical sabotage?